As we know, apples or mallow silvestris meal are one type of subtropical food that is widely grown in Indonesia. Apples are fruit that can grow at an altitude of 800 until 1000 meters above the sea levels. The productivity of apples in Malang has decreased drastically, and the severity of this is accompanied by a decrease in the quality of the apple produced. The poor apple that was once known to be fresh, sweet, and irresistible are now only underestimated by the customers. One of the factors that caused this is the attack of powdery mildew on apple plants in Malang. Powdery mildew or Lavella turica is one type of plant disease that usually attacks the leaf of plants, which are characterized by the presence of white spots on top of the leaf. The solutions commonly applied by the community to overcome these problems are spraying plants with nitrogen, potassium bicarbonate, or using neem oil. However, these solutions are still ineffective because they can lead to resistance to other plant diseases, have a negative impact on the environment, and also expensive. Based on this background, as a young generation, we feel responsible to bring back the glory of Malang's apple. Therefore, we are proud to present an alternative solution that is effective, efficient, and environmentally friendly with the title of The Utilization of Nano-Emulsion Bintaro Fruit Extract, Syahmina has as a fungicide exterminator for powdery mildew Odium vaninosum Cook in Malang apple plants, second year. I am Ardilla Syahmina, and this is my partner Awwaliyah Mahfudhoh from SMAN 7 Surabaya. Bintaro fruit is one of the most non-edible plants that grow in Indonesia. Bintaro plants are currently widely used as greeneries and decorate cities so that they have high productivity. However, so far, Bintaro fruit has not been widely used by the community because all parts of these plants are very poisonous. Bintaro contains alkaloid compounds such as polyphenols, saponins, tannins which are toxic, and contains heparin which can stop the heartbeat. Therefore, in our opinion, Bintaro fruit extract is very suitable if used as raw material for making biofungicide. To support the benefit and effectiveness of Bintaro extract, we utilize nanotechnology in the extraction process. Nanotechnology can reduce particles to nanosize so that the active ingredients are easily absorbed by target pests and make them more effective in eradicating them.
In the second year, we added the following tests: scanning electron microscope, gas chromatography, mass spectrometry, the test of the high pulse structure of powdery mildew, and powdery mildew resistant tests in apple plants before and after treatment of Bintaro fruit extract using the disease index scale. Civil Engineering Structure of Materials Laboratory of Brawijaya University, Chemical Laboratory of Malang State University, Advanced Materials Physics Laboratory of ITS, LIPI Chemical Research Center, and Bali Justru Batu City, with experimental methods and quantitative and qualitative descriptions. Tools and materials we use in this study are as follows. Stage of our research are as follows. First is the collection of raw materials, followed by pre-treatment of materials. Second, extraction process use a 19% methanol solvent. Third is sonication. Fourth is kept for 24 hours. Fifth is filtration use Birchner flask, and evaporation using rotary evaporator, and continued with PSA, SAM, QCMS, identification of powdery mildew high pulse structure, and resistance of powdery mildew to Bintaro fruit extracts. In the study, we used two samples, where all samples use a ratio of 16 grams per liter, but X1 samples without sonication process and X2 samples with sonication for 30 minutes. The results obtained from our research are as follows. First, each sample produces a different amount of extract yield, which the largest result is X2 of 5.5%. Second, particle size analyzer test showed that the X2 sample had the smallest size of 139.35 nanometers. Third, QCMS test showed that the X1 sample was thought to be dominant, containing 36.52% for nitropenyl laureth, which is antibacterial, antifungal, and antioxidant. 15.08% quinoline 3-carboxylic acid, which is antibacterial and antifungal, and 12.61% deutecanoic acid, which is antioxidant, antibacterial, antifungal, and antiviral. while the X2 sample is thought to be dominant, containing 23.03% for the propanil laureate, which is antibacterial, antifungal, and antioxidant, 20.77%, coumarin, which is antifungal, antibacterial, antiviral, anti-cancer, anticoagulant, and 15.32% dodecanoic acid, which is antioxidant, antibacterial, antifungal, antiviral. To ensure this dominant suspect compound is found in pinto fruit extract, we conducted an antifungal test obtained by the blend zone in the following X1 sample and X2 sample. For the powdery mildew resistant test against pinto fruit extract, we use 4 treatments, namely control, X1 sample, X2 sample, and X3 sample, which is a mixture of X1 sample with Nimrod 250 EC for 9 days, with a score using grid line and calculation of the following percentage of infection, then categorized into scale disease index. From the test of the resistance of powdery mildew to pinto fruit extract, obtain the results that X2 sample treatment is most effective in eradicating powdery mildew. The conclusion of this study are: first, pinto fruit extract has mechanism of work involving secondary metabolite compounds to inhibit the growth of powdery mildew. Second, the process of extracting pinto fruit with sonication process can increase the yield of pinto fruit extract so as to increase effectiveness in eradicating powdery mildew. Third, some tests and powdery mildew resistant test to pinto fruit extract will continue until the end of March to see the topography of pinto fruit extract particles and maximum ability of pinto fruit extract to eradicating powdery mildew.